Hello, we are Team 2A, Group 2, and this is our bioreactor experiment. E. coli is a naturally occurring bacteria in the intestines of humans and animals. Most E. coli strains are harmless, but others are pathogenic, which causes infections and illnesses. It is important to understand the growth of E. coli at certain conditions to aid in the treatment and prevention of infections. This figure gives a typical bacteria growth curve cultured in media. This figure illustrates the lag, exponential or log, stationary, and death phases of bacteria growth. The kinetic parameters for bacteria growth can be derived from this curve. In this study, the optical bacterial density will be measured over time, which will be used to solve for the biomass concentration. The kinetic parameters mu max and Ks will be solved for using equations 1, 2, and 3. For the LB media calculations, equation 1 can be simplified to the resulting equation shown. X and X naught are obtained by the measured optical density of bacteria. For the LB media calculations, the assumption was made that the substrate concentration is constant because the media is extremely rich in nutrients. From this assumption, the Monod constant, Ks, cannot be determined for the LB data. The maximum growth rate parameter, mu max, was determined from the semi-log plot of biomass concentration over time and was assumed to be equal to mu during the log phase of the plot. For the M9 media, the optical density was used to solve for x by equation 4. It was assumed that k was equal to 0.47 for this equation. 1 over s versus 1 over mu was plotted, and the slope and y-intercept were found using equation 5. Ks and mu max were then solved for using the relationships in equation 5. Since E. coli is a biological hazard, all work possible should be done in the biosafety cabinet. Even gloved hands should be sprayed with ethanol before using the biosafety cabinet to avoid contamination. All glassware and other equipment used for this experiment was washed thoroughly with soap and water. After cleaning, all openings were covered with aluminum foil and heat-sensitive tape was placed on each item. All pieces were then put in the autoclave to be sterilized. The pH probe was then calibrated. 2 liters of LB media was poured into the bioreactor. The pH probe and DO probe were then inserted and the DO probe was calibrated. Both probes were then connected to the bioreactor. Before sample preparation, make sure all flasks are properly labeled. 60 milliliters of LB media and 0.5 milliliters of E. coli were put into each of the 4 flasks. The benchtop shaker was set to 37 degrees Celsius and 200 RPM and left for 15 hours to inoculate. A blank sample of LV media was drawn and run in the spectrophotometer. Waste was put in the appropriate container. 5 milliliters of the inoculated bacteria was added to each of the 3 flasks containing 200 milliliters of fresh LB media. A sample was drawn immediately after the bacteria was added to the media to serve as an initial reading. 50 milliliters of the inoculated bacteria was added to the bioreactor. Every 30 minutes for 8 hours, a sample must be taken from the flasks in the benchtop shaker. Remove the samples and make sure to close the lid of the shaker. Bring the samples into the biosafety cabinet. Micropipette 1 milliliter of each sample into a cuvette. Be sure to change the pipette tip between every sample. After all 3 samples have been taken, be sure to place the aluminum foil back on each flask. Carry all 3 flasks over to the benchtop shaker. Be sure to firmly put each flask on the shaker. Close the benchtop shaker. Take a reading of each sample in the spectrophotometer. Record the reading in your lab notebook and dispose of the sample in the appropriate ethanol waste container. Draw out a 1 milliliter sample from the bioreactor using a syringe. Eject the sample into a cuvette. Remember to cover the bioreactor. Put the sample in the spectrophotometer and take a reading. Record the reading in your lab notebook, along with the reading for the pH and DO. Dispose of the sample in the appropriate ethanol waste container. For cleanup, place detergent in every flask that has E. coli. To disassemble the bioreactor, start by taking off the motor. Then disconnect the temperature probe, followed by the pH probe and DO probes. Remove both the pH and DO probes and spray down with ethanol. Place all bioreactor parts and other glassware used in this experiment into a basin containing detergent. Pour detergent into the bioreactor in order to kill the E. coli. Be sure to wash all other glassware and equipment used. Figure 1 shows the log and stationary phases of the E. coli growth in the bioreactor with LB media. One reason the bacteria may have stopped growing so quickly was there was a limited amount of dissolved oxygen in the bioreactor. The lag phase of the bacteria growth is not seen due to inoculating the bacteria 15 hours prior to the experiment. The experiment was not run long enough to see the death phase occur. From performing a linear regression on the log phase of this bacteria growth curve, the maximum growth rate, mu-max, was found. Figure 2 shows the log and stationary phases of the E. coli growth in the benchtop shaker with LB media. Only these phases are seen for the same reasons as previously stated. The maximum growth rate parameter, mu-max, for the benchtop shaker flask can be found in the same manner as done for the bioreactor data. As seen in Table 1, the flasks in the benchtop shaker took much longer to reach the stationary phase than the bioreactor. A reason for this can be explained by the amount of oxygen available to the bacteria. In the benchtop shaker flasks, oxygen would enter every time the cover was taken off the flasks, which happened every half an hour in order to take a sample. This was not the case for the bioreactor. The bioreactor sample was taken with a syringe in a way that very little oxygen could enter the system. Figure 3 shows the E. coli growth curve for the bioreactor in M9 media. Data was provided to us in order to perform this analysis. In order to calculate the kinetic parameters for the M9 media, 1 over S versus 1 over mu was plotted as shown in Figure 4. Utilizing the equation shown in the theory section, Ks and mu-max was calculated from the slope and y-intercept of this graph. The data shown in Table 2 does not correlate with what would be expected for E. coli cultured in M9 media. Since M9 media is not as rich in nutrients as the LB media, it is expected that as the substrate concentration decreases, the growth rate should decrease as well. In conclusion, this study explored the growth of E. coli in different environments. For the cultures in LB media, it was found that the maximum growth rate parameter, mu-max, was greater for the bioreactor when compared to the benchtop shaker flasks. This is what was expected because in the bioreactor, the E. coli has more contact area with oxygen in the air above it, therefore allowing for faster overall growth. Also, the bioreactor reached the stationary phase quicker than the shaker flasks. This is most likely caused by the dissolved oxygen being the limiting reactant prohibiting the E. coli growth. In the future, it would be recommended to control the dissolved oxygen percentage in the bioreactor so the data can better resemble the benchtop shaker. The M9 media data could not be directly compared to the LB media data or kinetic parameters because it had a stir rate of 500 RPM, while the LB media experiments were run at 200 RPM. The M9 data that was given did not follow the hypothesized trend that growth rate would decrease as substrate concentration decreased. It is recommended that M9 media data that is given have the same operating conditions so a direct comparison can be made between the LB and M9 media when culturing E. coli in a bioreactor.